Welcome to Learning Management System, Commissionerate of Collegiate Education, Vijayawada, Andhra Pradesh. Myself, Indravati, Assistant Professor in Biotechnology, KVR Government College for Women, Autonomous, Kurnool. Dear learners, the subject Recombinant DNA Technology is made easy for you. Do follow all my video lessons to cover different topics related to this subject. Dear learners, today we will be learning about ligation, mechanism and different types of ligation. So in this session you will be learning in detail about DNA ligases, the significance of these enzymes, the mechanism of ligation, the types of ligation and usage of linkers and adapters in blunt-end ligation. Let's study about the ligation reactions in detail. So, what are these DNA ligases? Simply, these ligases, they act like gum. You know, glue or gum, it is used to join the things. In the same way, the DNA ligases, they are used to join the DNA fragments. These enzymes were first identified in the year 1967 by scientists Marty Gellert, Hurwitz and Lehman. So these enzymes, they help in joining the DNA fragments. So in this example here you are having two molecules, molecule A and molecule B. So whenever they are digested with the same type of restriction enzyme, the BAM-H1, so you are getting two fragments having the sticky ends. So once you mix the fragments, they will be showing this transient base pairing, but the gap is there, that is discontinuity is there in the upper and lower strand between the first and the second bit GG, and here in the lower strand also. This gap is due to the absence of phosphodiester bond. So this gap, it is sealed by means of this DNA ligase. So the DNA ligase is used to seal the gap which is formed due to the absence of the phosphodiester bond. So when you are adding this enzyme, by means of the formation of this phosphodiester bond, the DNA fragments, they are joined. So biological significance, coming to the biological significance, these enzymes, they are mainly they are helpful during replication or during recombination and repair. Wherever the phosphodiester bond is lacking, there the discontinuities or they are formed, and these discontinuities are repaired by means of this DNA ligase. Whereas in commercial purposes, they are generally used to seal this blunt-ended and sticky-ended fragments in genetic engineering experiments. So wherever there is a discontinuity is there, those discontinuity is formed due to the absence of the phosphodiester bond, and those gaps, they are sealed by means of this DNA ligases. Coming to the basic reaction, DNA ligase reaction. So by means, that is there are two fragments, first fragment and the second fragment. So these two strands can be binded. So the 3' hydroxyl of one strand, it binds to the 5' phosphate of the other strand. By means of the phosphodiester bond formation, these two strands, they are able to ligate, and it is a highly energy consuming reaction. For this, it requires ATP or NAD+. The mechanism of the reaction, it is explained in detail in this picture. So this brown color one is the DNA ligase enzyme, and this enzyme, in the active site of this enzyme, it is having an amino acid called as lysine. So when this cofactor ATP, it is broken down to AMP and pyrophosphate, PPI, pyrophosphate, the AMP, it goes and binds to the active site, that is to the lysine, by means of phosphoamide bond to form enzyme AMP complex. So the first step, it involves the formation of enzyme AMP complex. This complex then binds to the 5' phosphate of the DNA fragment and it activates the 5' phosphate. The enzyme complex activates the 5' phosphate in the second step. In the third step, the hydroxyl group which is present in the adjacent strand, it attacks the activated phosphate and a phosphodiester bond is formed. And at the end, the AMP is released. So these are the three steps. That is, the first one is the enzyme AMP complex formation. The second step is the activation of the phosphate group by the complex, and the third step is the attack of this 3' hydroxyl group to the activated phosphate group present by the formation of this phosphodiester bond. At the end, AMP is released. So during the reaction, two high energy phosphate bonds they are utilized. Commercially, these DNA ligases are extracted from T4 bacteriophage and E. coli. So whatever may be the source, the mechanism of action will be the same. That is the formation of phosphodiester bond. They will be joining the DNA fragments. These T4 DNA ligase, they will be differing in the cofactor requirement. They require magnesium and ATP, whereas E. coli DNA ligase, they require magnesium and NAD plus as cofactors for successful completion of the reaction. And also this the T4 DNA ligase, it can promote both blunt end ligation and sticky end ligation, whereas E. coli DNA ligase is used exclusively for promoting this cohesive end ligation or sticky end ligation. So that is the difference. So this blunt end ligation means that is when the restriction fragments they are having these blunt ends or fleshy ends, then this T4 DNA ligase it is used, whereas the sticky ends will be having this single stranded overhangs. So before ligation itself, this transient base pairing will be occurring and it will be stabilizing this particular DNA strand, and further by formation of this phosphodiester bond by the DNA ligase, the sticky end ligation is more efficient compared to that of the blunt end ligation. So that is what is represented in this slide. The specificity of the restriction enzyme and also the hydrogen bonding of this cohesive ends. This hydrogen bonding is occurring prior to ligation because due to complementarity in the this overhangs, the transient base pairing will be occurring and it will be further stabilizing this cohesive end ligation, whereas in the blunt ends because the ends are fleshy and you are not having this overhangs, this type of blunt end ligation is less efficient compared to that of the cohesive end ligation. So in order to overcome this particular limitation, in order to increase the efficiency of blunt end ligation, you will be using certain synthetic molecules, oligonucleotides like linkers and adapters. So these linkers and adapters, they are the short DNA fragments which are attached to this blunt ended fragments to convert this blunt ended DNA molecules to sticky ended form. So linkers are short DNA fragments which are having internal restriction sites within them. So in the example here, this is the BAM-H1 linker. This BAM-H1 linker is containing BAM-H1 restriction site. So this restriction site is present internally in the linker molecule. So these linkers, they are added on either side of the blunt ended molecule. And by means of the DNA ligase, these linkers are attached on either side of this blunt ended DNA. After attaching, the linkers when they are subjected to this restriction enzyme digestion, BAM-H1 is added. So this bomb H1 will be because it is having the recognition site within the linker, it will be cutting at the ends, and in this manner you will be getting sticky ends at the ends of this fragment. So the linkers, they are used to convert blunt ended fragments to sticky ended fragments. How they are formed means because they are containing internal restriction sites. Due to this restriction sites, whenever you are again digesting with the restriction enzymes, the blunt ended fragments will be converted to sticky ended fragments. So they are exclusively used in gene cloning experiment to insert the target DNA into the vector molecule. So this is the target DNA molecule. To this, on either side of this target DNA, the BAM H1 linker it is added. After that, this target DNA is cleaved with the BAM H1 restriction enzyme. So you will be getting this type of 5' overhangs or sticky ends on either side of the target DNA. Then the vector molecule will also be digested with the same BAM H1 restriction enzyme. So this also will be containing the overhangs which are complementary to that of the target DNA. So whenever you mix the target DNA and the vector molecule, due to the complementarity, both will be joining, and you will be seeing the formation of a recombinant vector. In this manner, for the creation of a recombinant vector or recombinant DNA, linkers play a very important role. The second one is adapter. So adapter is also a short DNA fragment which is also having restriction sites within them. The difference between linker and adapter is linker is having it is blunt ended at both the sides, whereas in adapter the one end of the fragment it is blunt, whereas the other end it is having preformed sticky ends. The other end is having overhangs. So that is the difference. So these adapters, they are attached to the target DNA molecule, and after that one, this another important feature is the 5' end of this particular adapter molecule is lacking the phosphate group. Here you are seeing it's modified, it is having hydroxyl group. So why this hydroxyl group is added means is why the modification means in order to prevent self ligation of the adapters. So whenever the phosphate group is present, so there is a chance that the adapters they ligate themselves, and in order to prevent the dimer formation, you are using modifying the 5' end with this hydroxyl group. So that is also one of the thing to be noted. So this is the target DNA molecule, the blunt ended target DNA molecule. On either side of it, you are adding this adapters by using the enzyme DNA ligase. So this target to this target DNA, the adapters will be going and joining, and because they are having the preformed sticky ends, ultimately our blunt ended fragment will be converted into sticky ended fragment, and the modification, that is you are having this hydroxyl group in this the terminal end, that one it is removed by using the enzyme kinase, polynucleotide kinase. When this enzyme is added, the hydroxyl group is removed, and in that position the phosphate group is added. So in this manner you will be getting that is the blunt ended fragment is converted to a sticky ended fragment by the usage of adapters, and also these they act in the way similar to that of the linkers, and they are used in gene cloning experiments. So conclusion, these ligases, they help in the joining of the DNA fragments by the formation of phosphodiester bond between 3' OH and 5' phosphate group, and of the two types of ligations which we have discussed, cohesive end ligation is more efficient due to transient base pairing of the overhangs, and the blunt end ligation is less efficient, but it can be overcome by using linkers and adapters. The linkers and adapters are these short DNA fragments which are containing internal restriction sites and helps in the conversion of blunt ended fragments to sticky ended fragments. With this, I conclude the session. Hope you enjoyed learning this session. Last but not the least, I profusely thank Commissionerate of Collegiate Education for providing this LMS platform to reach a large number of enthusiastic learners like you. Thank you one and all.